Good evening, ladies and gentlemen. Yeah, okay, there's at least a few. We're spread out. My name's Kent Spears. I'm a very part-time research assistant at the Arctic Institute. I also do social media, so I'll be tweeting some photos of our presentation tonight. This is our first speaker series of this academic year. We've changed things up a little bit. Our start time is a little bit later to see how that would work for folks. And typically, we've invited people back to the Institute after the talk, but we've changed it up this year a little bit. So we're inviting people to the Last Defense Lounge, which is on the second floor of the MacEwan Student Center. So after our talk, you can join Rhonda there, and you can buy her a beer or two and ask her some really in-depth, tough technical questions that myself as a social scientist would never ask. Okay, so I'm happy to present Rhonda Clark. Rhonda joined the Energy, Bioengineering, and Geomicrobiology group in the fall of 2016 as a research project coordinator for the Geomicrobiology group and project manager of the Genome Canada GENICE project awarded to Casey Hubert. Prior to joining EBG, she worked with Dr. Garrett Verdow at the Petroleomics group here at the university for over 8 years. From 2002 to 2008, she was a sessional instructor in the Department of Biological Sciences, teaching microbiology, and a research associate working on the genetics of nitrogen bacteria. Rhonda's PhD from the University of Alberta work focused on the rumen bacterium in cows and reindeer, developing novel genetic transfer systems that would allow for the introduction of genes involved in the cellulose degradation. This work was done at the Rowett Research Institute in Aberdeen, Scotland, the Agricultural Canada Research Centre in Lethbridge, and the University of Calgary. Upon completing her PhD, she was presented with a very interesting job opportunity that involved coordinating the 100th anniversary commemorative cattle drive that took place at CFB Suffield and ended in Medicine Hat, Alberta. Very true Albertan project, that's great. The work involved acquiring sponsorship, arranging contracts and service providers, meeting with stakeholders, tracking participant registration, and looking after finances. So Rhonda is excited to be supporting EBG in a position that utilizes not only her experiences and research, but also her project coordination. So please join me in welcoming Rhonda.
Well, thank you, Kent. And I feel no pressure because my previous boss and my current boss are both sitting here in the crowd. Anyways, thank you for the opportunity, and I'm glad to see some people came out to listen about microbes. Maybe not the most interesting topic to a lot of people. But so my title of my talk is Microbial World in the Arctic, Baselines and First Responders. So we have this project where we're in the Arctic and we're trying to identify the microbial community that exists in an undisturbed environment. So that's our baseline. And then ones that are in that community that may potentially degrade oil. So our first responders. Now, to do research, you need money. And so we've been fortunate that Casey was awarded Genome Canada large-scale applied research program. And we call that GENICE, so Microbial Genomics for Oil Spill Preparedness in Canada's Arctic Marine Environment. It's supported by, of course, the Genome Canada and some of the associated regional genome centers and Research Manitoba, which, because we have partners at these institutions, University of Calgary, University of Manitoba, and McGill is where our research teams are centered. And then, of course, we have co-funders, people who provide in-kind co-funding so that we can achieve all the objectives of the project. It's like it's on hyper forward. So, like I said, the research teams, we work in these 3 institutions, and we have a broad range of skills that we bring to the project. Our group works on the genomics, but this is a gathering of a number of us from the 3 centers in Winnipeg in the spring for a meeting and I'll talk about that meeting later. And then this is our geomicrobiology group here at the University of Calgary, the group I work in with Casey. And so there's a few of us in this group that work specifically on the GENICE project. So what do we know about microbes and oil spills? Well, we actually know quite a lot. The one that most people, or oil spill that most people will think of, is the Deepwater Horizon or the Macondo well blowout in the Gulf of Mexico. And so that happened in 2010. And so in the past 8 years, there's been extensive research done on the microbes that activated and started eating the oil that came from the well. So these are just, I actually stole this slide from Casey. I like it that much. So, just different articles, bacteria feasted on Macondo methane, an explosion in oil munching bacteria. There's this one down here. I think this is the one, if you go, they've got a little slideshow of the different microscopy of some of the different microbes that they identified. So it's kind of neat. So we know lots from that. We know that, I don't know why this is doing this. We know there's bacteria that eat oil. So I'm going to use oil and hydrocarbons interchangeably, and why we call petroleum products hydrocarbons is they're made up predominantly of hydrogen and carbon, put together in different combinations that will give you some of the different petroleum products. And so it's all these different chain lengths. Some of them are more easily eaten by bacteria versus ones like naphthalenes and paraffins. They're a little more recalcitrant to being degraded by bacteria. And so when we say they eat oil, basically they use it in their metabolism. So they need food like we do. So they'll take this hydrocarbon as food. So we've got that there. And bacteria that grow in the presence of oxygen can take this hydrocarbon complex together with oxygen, and it helps them create energy, and they can create carbon dioxide and water. And there's also bacteria that can grow without oxygen. And to do that, they'll pair the degradation of the hydrocarbon with reduction of something like sulfate. And in that case, you'll get CO2, but you'll get hydrogen sulfide. So that's how, when you talk about eating oil, that's what they're doing. So there's a lot of learnings from the Gulf of Mexico and Deepwater Horizon, but of course an oil spill in the Arctic intuitively will be different. You've got ice conditions, you've got different temperatures in the Gulf of Mexico, and so there's a whole different set of parameters going on in this environment. And so our project is studying the microbes in ice and water and sediments in the Arctic. Keep going over here. This is like a really hyper, it's like I just have to look at it. So in the Arctic, people might think of oil production, offshore rigs, but there's not really any oil production going on in the Arctic yet. But what you are seeing is more passenger and cruise ships going through the Arctic, resupply ships bringing in diesel and supplies into the communities. And I pulled this one. This was actually a grounding of this ship, Academic. I don't even know how to pronounce that, but basically it grounded, and nothing happened. There was no fuel spill or anything like that. But these sorts of incidents are potential, right, for hydrocarbon fuel spills in this environment. So how do we go about studying all of this? Well, we're very fortunate in our project to have access to much of the Canadian Arctic. We focus on the Hudson Bay region in here, and then the Northwest Passage, and then up in the Baffin Bay area. So this is an example from this year's research expedition on the Amundsen, where this is basically what they lay out as the research cruise track for the summer. And then we have the opportunity to go on the ship and sample at all these stations if we have people on the ship. And so this is the Amundsen here. So this is the one, probably we've done the majority of our sampling from the Amundsen over the last few years. And then there's the Arctic Research Foundation has a couple ships. This is one Srijak, who's here, he and one of our grad students went on the William Kennedy this summer from Prince Edward Island. And well, they were supposed to go through the Northwest Passage, but made it to Nain. But anyway, so we have access to these ships, and we can go out and we can get our samples. So the samples we want to collect while we're out there, first there's sediment samples that we want to collect. So we have, this is on the Amundsen, all these operations are on the Amundsen. So this is what we call a box core. And we're able to take this box core and with the help of trained crew on the ship, it goes on a winch and then it gets lowered down to the seafloor where it can scoop up sediment and then bring it back up on deck. And then we get to process these sediments. So here's what it'll look like when it comes up. And so it's a bunch of mud, and then we can take these push cores and we push them down in. And can you guys see that okay? Or should I turn off? Which one? Bulkhead? Nope. Maybe those ones. Maybe we just do all right. Whoa, that's too dark. That's better. Is that better? Okay. Okay, so there's our sediment or mud that comes up, and we do the push core, and then it'll come out like this. This is one of our postdocs, Anirban, who was out on the ship this summer. And so we've got this nice push core, and then we can take it back to our outdoor lab, and we can start taking samples at the different depth levels and preserving those for future analysis. And then we also like to collect the seawater, and so we use a CTD rosette. So the rosette, the CTD, it's taking conductivity, temperature, and depth as it goes down. And this rosette has a bunch of these little, what we call Niskin bottles all around. And then we get assigned a certain number of those bottles, and we tell the rosette operator, who's in a little room up in here, that we want water from 10 meters below, maybe halfway down the water column and close to the bottom. And they can open up, Niskin 1 for us just below surface, Niskin 2 a little further down. And then that'll come back up to the surface. And then you go into the rosette shack inside where it's warm. And then you start collecting your water out of those and into your containers. So you can also take those back to process and preserve. And then we have this really highly technical operation for collecting surface water. It's called a Home Depot bucket and a rope. And we just toss it over the side of the ship and we collect our surface water. There's not too much involved, except there are certain techniques that work better than others for getting it to deploy and go sideways properly. So being a true Albertan, I had to show the team last year, don't just flop it, you should actually do this. And then, anyway, that was you have to have fun when you're out there. This year, after, I think this was attempt number 3, is that right, Casey? We were able to use a remote operated vehicle and get very targeted samples in the Scott Inlet region of Baffin Bay. And in this region is an actual natural hydrocarbon seep, so where hydrocarbons are seeping from below surface. And so this was targeted because we want to go in and specifically sample in an area that we know there will probably be bacteria that are already adapted to hydrocarbon degradation. So attempt number 3 this year finally worked. The team was able to go in, the Amundsen was able to go in. They were able to deploy the ROV. And so we got 3 short ROV dives. And then there were plans to do, the box coring, but the bottom was quite rocky. So that's not really possible because you'll wreck the box core device. So they replaced it with a water sampling in this transect from the hydrocarbon seep and then out away from it in different directions. And then all those samples are coming back and we're going to start analyzing those too. And so this is just some pictures of the team in the ROV room on the Amundsen. And so here they're looking pretty relaxed, and here they're pretty intense because now like things are really in action, and they've got to watch and make sure they've got it in the right area and that they're ready to grab samples. So these are just some of the pictures you can from when they had the ROV going down. So that's what the bottom looks like. And then this is how they grab the little, they do grab samples of the sediment instead of the box core. So we still get access to some sediment, just not a large amount. And it, well, what's so, and we also need sea ice samples. So I'm just walking you through. The pictures are more fun of collecting the samples. So to collect sea ice samples, you have two options from the Amundsen. You can, the ship can get in beside a good area of ice, and then they'll deploy the team down in this cage with all their supplies, and they'll go out onto the ice and start coring. Or they can go out with the helicopter, which is on the ship at all times, and get their ice cores and any other things they want, like melt pond water, things like that. And so we've collected all the samples. So we've got basically our time zero samples we've taken, and now we can take subsamples of those, and we put them into these little tiny serum bottles. We call them microcosms, or mini oil spills. Because what you can do is you can take your different samples that you've collected. So let's say in here, this example is sediment samples. And you put them into the bottles, and you can add diesel, or you can add bunker fuel, you can add crude oil, and then you have your controls which have nothing, and you're going to compare what's going on in the ones with the fuel compared to the ones that have none. So I'm going to talk more about these, but so that's our little microcosm experiments that helps us select out the ones in the community that may be able to degrade the hydrocarbons. And then this is how we deal with the water. You need to concentrate down the microbes that are in all that water you collect. And so we filter liters and liters of water through this vacuum manifold, and then we get the filters, and then we can get the DNA off of the filters with special kits. So we've kind of set up our samples, we've set up our microcosms, so now we want to characterize the microbial community and the activities. And so this is where the genomics comes in, which is what our group here in Calgary focuses on. And then University of Manitoba focuses more on the petroleomics, as well as the sea ice geophysics part of the project. So microbial genomics and all the other omics techniques give us the opportunity. By isolating DNA, we can figure out who's there. Now, they may not all be alive and functioning, so then you can go into the next step of transcriptomics and find out who's active in that community that you're analyzing. And we've got proteomics, figure out what are they doing, and then the metabolomics, what did they do? And then the petroleomics is a nice tie-in with our project because you want to figure out what parts of the hydrocarbon you've added to those microcosms is actually being degraded and at what rate. So it could be really fast, it could be really slow. Those are all really important things for us to find out. And it's doing it again. So, oh my God, put that down. So we use something called the aluminum MySeq, and that's our special little machine that we can take our samples, so whether it's that original sample or sample from the microcosm, and we can isolate the DNA, and we amplify that so we get more volume, it's more readable on the machine. We do some other fancy stuff before it goes onto the machine, and then it spits out this reams of data that you then feed into a computer program where we do bioinformatics. And it's that programming that tells us all of the different bacteria that we have in that sample of mud or that sample of water or in the sea ice. And so that's our genomics component and what we're working on. And then at Manitoba and a little bit here at University of Calgary and Steve Larder and Thomas Oldenburg's group, we do the Petroleomics. So then, and let's say from our microcosms, you can take samples and they can put it onto all their fancy equipment they have in this lab. And they generate these very interesting graphs that tell you how much of the hydrocarbon that you put in, how much has disappeared, what components have been eaten in the sample. And so that tells us the rate and the level of what's being degraded. Then we also have the sea ice geophysics team. So this is sort of not an omics part, but it's because we're studying ice, you need to have these techniques as well. So when they're out on the ship, they'll do things like use scatterometers or radar equipment, and they can look at quality of the ice, age of the ice, a lot of different parameters to help define the ice. And then these techniques are also very helpful when we come into a more controlled lab setting. So at Manitoba, there's something called the Sea Ice Experimental Research Facility. And so we can take these, basically they're big tubs, and they can put seawater in them. And this happens in the winter, of course, in Winnipeg, so you have to wait till probably like November, December. And then they can add in oil, so like a mock oil spill. And then they can use those scatterometers and radar equipment, and they can look at how the oil interacting with the ice is changing parameters of ice. I don't know ice geophysics very well, but look at that. And then we can go in and take ice cores. So I remember when Casey first saw this, he thought these were Cheerios floating around in the tank, but it's actually where they've done the ice core, and as it comes out, it puts ice over the side. And so you can take the ice core samples, they can do the petroleomics, and we can do the microbiology on those. And, oh my, Melanie, I don't know about this okay, it's staying. So out of the research that I've just explained, the outcome we're looking for is, through all the sampling area and hopefully over 2, 3, 4 years where we can get samples, we can establish a baseline in the shipping corridors where we're sampling, maybe close to communities. And so it's a baseline before an event has happened. And that can be used in something called environmental effects monitoring. So if there is a spill event, this gives you kind of what was the microbial community before the spill, and as you're doing all of your spill response strategies, are you achieving close to what was there before the spill? So it's a way to monitor, I guess, how effective spill response would be. And then another outcome we have from the genomic side is a microbial-based spill response strategies to give to, let's say, the Coast Guard. So in certain areas, are there bacteria that would become really active, like in the Gulf of Mexico, and be able to start acting as the first responders, because this is a remote environment. Remember, the Coast Guard has told us in some areas, Hudson Bay, it could take them 10 days to get to where a spill would happen. So this may be a way for them to know there's bacteria out there that are starting. They may be able to, when they get there, add nutrients or something that may speed that up, but also do their other spill response approaches. An exciting part of GENICE, I think, and I've learned a lot from this side of the project, is what they call the GELS. So GELS in a Genome Canada project stands for, it's an abbreviation for Ethical, Environmental, Economic, Legal, and Social Research. And that's something that Genome Canada, they always want as part of their projects. I think it's really great. We've had a really active GELS team since the beginning of the project. We're 2 years into the project, just about, technically. And so since about December 2016, we've done a lot of these things. We've had end user meetings. So we've met with community representatives, whether that's the mayor of Chesterfield Inlet or Churchill. We've had from the hunting and trapping associations, Coast Guard's been involved in meetings. And these have been I think in regards to what we do as you know, the natural sciences researchers, incredibly useful. So, you know, we went to Winnipeg, met with a lot of these end users, and we were all gung-ho about explaining genomics and what we're going to do with genomics. And we sat in all these breakout groups together, and we all came back after and did a report. And every single one of the community representatives that were there talked about, well, hey, we've got these spill kits in our community, and they're full of all this stuff. We have no idea what the stuff is for or how to use it. So that brings you back to a practical level of, they've got, you know, a really basic concern that needs to be addressed first before we come in with our big, I don't know, those technical approaches to spills. Coast Guard meeting was really awesome. We got together with different Coast Guard personnel, and we were talking about our little mini oil spills and all the crude oil we were adding and all of that. And they listened, and then they said, well, there's not really any oil exploration per se, and it's good to study that because in the future there might be. But really, what probably would be more useful right now is, what about ship diesel or bunker fuel, or the diesel that's going, like the heating diesel that's going into the communities? And it's like, oh yeah, that would be a better approach. So now we've modified our experiments to include those sorts of hydrocarbons. We've had a law and policy implications meeting. So a really interesting interaction that came out of that was, we were talking about our first responders, and the bacteria will look after, you know, maybe they'll look after the oil. But you have to be careful how you present that, because could that become something where, if it's not phrased properly, like monitored natural biodegradation, does that give an out to, let's say, a company that spills in like the Northwest Passage, go, well, we don't need to worry about it because the bacteria are out there. So it's just all these really, really great input from another side that has been very valuable to the project. We've had transportation and economics meeting, which is that picture I showed you back at the beginning. So that's, we met with, well, international and then of course University of Manitoba team to really understand all the data we're generating, how can that feed into their models that they do for transportation risk and assessment. Chesterfield Inlet community visit by one of our University of Manitoba GELS team members. There's been a ship visit by the community on the Amundsen when it was at port in Churchill in July. So these are all really great interactions. I think it gives a lot of strength to the project. I feel like it's a more robust and perhaps bring more relevance to what we're doing, as well as the natural scientists. So this is just, I'm going to end with a few pictures. This is a very short talk because you can only talk about bacteria so much for people want to leave. So there's the Amundsen. It stopped in Churchill, and this is the community visit where they came on to, people who were interested could come onto the ship and just see what goes on. And this is our very great summer student, Michael, who a few days after he started, we said, hey, you're going to go out on the ship for 6 weeks. And he did it. He did it, no problem. He was a star. And that's just him showing some of the people who were on the ship what we were doing in the lab. And this was pictures of the community visit that Glenn from University of Manitoba did. This is the mayor of Chesterfield Inlet, Simone. And looks like they're checking out all of the stuff that's in there. The one thing that Simone and the previous mayor of Chesterfield Inlet and Mike Spence from Churchill, you know, they all say people get trained on these kits when they first come into the community. That's the, like the sea cans there with the stuff. But then if that person moves on, that knowledge is all gone. So I guess that's a challenge probably that probably the GELS team will also work with them to make sure that gets communicated to different people within the community. So that's all I have to talk about tonight. And if there's any questions, we can chat that way. Thank you. You're awake, Melanie?
Rhonda, is it the Coast Guard that supplies the kits?
Yeah.
So they don't do any follow-up?
Well, I mean, they're a pretty big organization. When we were in Halifax for the law and policy meeting, there was actually some Coast Guard people there, some of the operations people, and we mentioned that again to them, and they were really surprised to hear that, and they put it on like an action item. So, I mean, hopefully, but I guess it has to work its way down. But they weren't really aware that, but yeah, it's those practical things, right, that I think we get all up here, and it's like, oh yeah, well, what do you do with those hoses? I don't know, that were in there.
Are the Russians pumping oil on their side?
I can't answer that. Yeah. What do you think? Can you, Casey?
They aren't already. They're much closer to. Or much closer to it. I don't think
This is my boss, Casey.
so Obama and Trudeau signed an agreement not to be only looking for 5 years. Trump may disagree with that, but certainly in Canada we don't do oil exploration in the Arctic right now. But I think in Russia for sure, that's happening. I don't know if there's active production though. I'm not sure about that.
Pretty darn close to it, they're not doing it.
Yeah, definitely. It's a different posture.
Oh, yeah, much.
We can say that confidently.
You had a question, Shreya?
I don't know, I just
Oh.
wanted to add that the newest plan that the government report came up with includes exploration areas which are basically in North Arctic Alaska, adjacent to the Canadian border. So, given how the ocean currents are, even if they explore and create a spill, that is not to say it won't go into our water.
Matt.
I know it's studying the diagram of all the sea ice. It had oil coming up into the brine channels.
Oh, yep.
Does that happen during formation, or is that after?
You're asking all the tough questions. I don't know if it comes up during the formation of the brine channels. Do you remember what they talked about?
Sorry, how would the oil get into the brine channels?
Yeah, like is it as the sea's freezing, and it's freezing the oil?
It would have to, I mean.
Or would it get sucked up after the brine?
So as Rhonda mentioned, we do the sea ice geophysics experts that probably know.
Yeah, you probably know them. Okay.
And those tanks with Cheerios on top, so that would be an opportunity to do an experiment to see how the oil behaves in the ice. And what's quite interesting in the brine channels is that, you can maintain liquid brine down to -20. So could you have an organism that can live at -20 in liquid? So liquid water principle is possible. So -20
And
really
saline.
salty
Yeah.
and still like to be oil. That's a really obscure organism, but our rule of thumb is most things exist in microbiology. So
You found any of these organisms that people oil in the Arctic yet?
Oh, yeah. Yep. So we've got one of our former grad students did a lot of that sampling, and she's like, they've identified organisms that eat oil. So, yeah.
Is there a bit of a record of kind of spills that have happened? Of course, typically the spills, the very large ones, the huge ones that you mentioned, they are devastating, but they
Yeah.
can be, but they are pretty rare.
Yeah.
Some are, once every 20 or 30 years, small spills where, ship.
Well, yeah, they don't get the.
And nobody is any wiser.
Mm-hmm. Yeah, and it does happen more frequently. So just, they talked to the Chesterfield Inlet representatives talking about, they've had, when they're offloading the diesel or fuel to go up to the mines, they'll have spills like in close to town. When I was getting off the Amundsen in Frobisher Bay, there was a big diesel tanker came in, and so they actually resupply out in the bay, right? And there's hookups where it comes off the ship. So yeah, those kind of spills are probably the more likely ones. So not the big massive spills, and then maybe like a passenger ship runs aground and damages.
But also, you talk about heating fuel, the communities are fairly small, I mean, 500 people is fairly typical.
Well,
You know,
Iqaluit's about 8,000 people, so
Yeah.
they've got quite a big tank farm.
uh,
But yeah, the smaller ones. But I mean, it's still, you know, even a somewhat small spill in, let's say, a bay, you know, it can affect, you know, their fishing or that kind of stuff.
Do they
So.
have very good reporting, you think? I mean, it's such a remote area.
Yeah, that I don't know, like how they report a small
Something
spill.
close to Vancouver, you
Oh yeah,
need
totally.
to-
I think, like, we've had an opportunity, Simone and then Barney, who was the mayor before, and Mike Spence has been at a lot of our meetings, and I think they're much more on top of that stuff now. I mean, they welcome some of the development in because it brings jobs, but there's also that other side of you know, pollution, and so they probably are a little more on top of it, monitoring. Now, I would suspect be communicated back to the Coast Guard. But I mean, the Coast Guard's so far away. I guess that's why they put the spill kits in the communities and hope that they can, the spills are small enough that they can do that physical response. Yeah.